Good morning, everyone. We start our lesson. I hope you have good mood and you prepare very well for our lesson. And in the first of all, I want to say about your homework. We want to check your knowledge, how you understood topic which we studied yesterday. Okay, guys. Now I sent a link in our chat. Please open this link. It's Padlet. When you open this link, you can see some questions. You can see some questions about competitive and non-competitive inhibitors. Here we write your name and questions for you. Okay, guys, you have just 5 minutes for writing your answer. Please write your answer. If you have some problems, you can say, for example, you cannot write or another problem. Guys, do you have some problem?
And since this is a revision, okay, try to answer the questions without looking at your notes, okay? So just answer it with the knowledge that you have gained or the things that you have remembered from our previous lesson.
Yes. Guys, 5 minutes. Now you have 4 minutes. Now I can see just Alpamis wrote.
Yes. So far it's only Alpamas who have answered the question. Mohamed Yar, Medina, Abdul Kahur, Shakruk, Mahinur, Ruslan. You have 3 minutes. Okay, so Mohamed Yar also answered the question. Okay, the competitive inhibitor binds to the active site and prevents the substrate from binding there. Okay, that's correct. The non-competitive inhibitor binds to a different site on the enzyme. What do you call that other site other than the active site? There is a term for that, Mohamed Yar. It also starts with A. It's not an active site. Any other site than the active site is known as, if you would just look at the task of your classmates, it's there. That's actually assigned to one of your classmates. But yeah, you're correct about the difference between competitive and non-competitive.
And Alpamis correct too.
Okay, Alpamas, when you change the amount of enzyme, you change the Vmax. So in the presence of a non-competitive inhibitor, the Vmax decreases, yes. The fixed amount of inactive enzyme in non-competitive inhibition does not affect the Km, and the Km is not changing, or it could even be lower, okay? Now, how about in the presence of a competitive inhibitor, Alpamas? Because you only put here non-competitive. What happens to the Vmax and the Km in a competitive inhibition? Mahinur. Okay.
It will increase.
Okay, which one will increase? Km or Vmax?
Vmax.
Vmax is Km. Km will increase, okay? But Vmax will still be the same, okay? It will still reach the same Vmax as long as you increase the substrate concentration. So could you please put that in your answer? Add that in your answer.
Okay, Medina gave answer too.
Okay, could you please read Oxy? What did Medina write?
Set a site that allows molecules to either active or inhibit. Yes, it's correct. Your answer is correct, Shahrukh.
What is a Michaelis-Menten constant? It's the affinity, yes, of an enzyme for its substrate is most commonly measured using the apparent Km, which can be defined most simply as the amount of substrate necessary to allow an enzyme to function at half its maximal velocity. So in a graph, you can see the Km by looking at the substrate concentration at 1/2 of the Vmax. So that's correct. Okay, so it's a measure of the affinity. Okay, how good the substrate is how good the substrate in being attracted to the enzyme. Okay,
Ruslan is
Mahinur.
correct.
Ruslan is also correct.
Yeah.
X is normal, it's competitive, and Z is non-competitive because Z doesn't reach the same Vmax. Y, it has a lower rate but can still reach the same Vmax. Okay, and obviously X is normal. And then Mahinur, malonate binds to the active site of the enzyme without reacting. And so competes, not completes, okay? Competes with succinate, it decreases cellular respiration. Very good, okay? So malonate is a competitive inhibitor to the enzyme succinate dehydrogenase. Succinate is the normal substrate. That's why she wrote here, it competes with succinate, which is the normal substrate. And if succinate is not bound to the enzyme succinate dehydrogenase, the cellular respiration or the rate of the cellular respiration will decrease. Very good.
Okay,
Thank
but
you.
we don't have answer from Abdukahor. Abdukahor. Abdukahor.
Yes, yes.
Where is
Okay.
your answer?
So it has something to do with whether the inhibitor binds permanently or temporarily to the enzyme. Okay, so think of non-competitive inhibitor. Does it bind permanently or temporarily? So that's the reason why it levels off sooner than a competitive inhibitor. So imagine you have 10 working enzymes and then 5 of them are in non-competitively. So you only have 5 working enzymes left. So that's the reason why it cannot reach the same Vmax. Okay, so that's a hint.
Okay.
2 minutes. Yes, Oksana.
We finished, yes, with check knowledge, with the revision.
Abdul, do you still want to put your answer? You have 2 minutes.
Yes, we will waiting him.
Okay, wait for his answer.
Abdukahor, you can write or not?
He can.
Abdukahor.
1 minute, please.
Okay, all right. Go. It's okay. All right, now, after reviewing the previous learning, so you guys have to go to the group work. So I already showed that to you before. So in Teams, there is a tab there called group work. Now I want you to find the page that is assigned to you. So you will be working in pairs. So there is a reading material or a text that you need to, of course, read and understand. And then there are 3 things that you need to identify in the text. I hope that you guys are listening. So first is the name of the inhibitor. Second is how does the inhibitor work? And third is the effect. Okay? So in identifying, you just need to highlight these 3 things within the text using different colors. So please, for those who are already done, please go to the group work tab in Teams and then find your name, find your page, and then start working on it. So these are the positive and the negative effects of inhibitors. So you have 5 minutes to do that. 5 minutes. Okay, so I'm going to share my screen, Oxy. Is that okay?
Okay, Abdulkahor wrote answer.
Okay, so while Abdul is writing his answer, so let me just share my screen with you so that I could also see the progress
Yeah, sure.
of the work of the students. Okay, so go to Teams. Okay, and then you have group work tab. All right, so just find your names. For example, group 1 is Ruslan and Shakruk. So your topic is about snake venom. Okay.
Okay, I've seen it.
So highlight the part that says the inhibitor name with green. Okay, how does the inhibitor work with yellow and effect blue? Okay, so you can use the highlighter. Then Abdukuhur Alpamas, you have cyanide poisoning. So just the same, identify the inhibitor name, how does the inhibitor work and the effect using the designated color of the highlighter. Okay, 3 minutes. It's a short text. And then Medina, yes, you have to work.
Leo, I have
Yeah.
some problems when I group work touch. They need download.
But can you see the text? Here's their text.
Medina, can you see the
screen.
text?
No, я когда нажимаю на group work, там просит у меня скачать приложение.
Okay, but you can see my screen now. You can see my screen now.
Yes.
Okay, now this is the text assigned to you. So I will let you read it.
Okay.
Okay, and
Okay,
then
it's
identify, just tell me the inhibitor name, how does the inhibitor work, and then the effect. And
Okay.
then we will look at the other students' work after. Okay, 2 minutes.
Оксана Руслановна, Алпамус не может зайти в этот групп орг.
Alpamas, which problem do you have? Alpamas.
У меня экран черный стал. Сейчас надо Wi-Fi переподключить.
Okay, Mr. Leo.
Yes.
Almas has some problem with internet connection.
Okay, so Medina, are you done reading? Medina? 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 Okay, so in this text, it's called suspected antifreeze poisoning treatment. Okay, so it says here that ethylene glycol could be a potential poison and can be ingested accidentally. Okay, so if this chemical is ingested, there is an enzyme in the cell, in the liver, that can convert it into oxalic acid. Okay, and oxalic acid can lead to death. Okay, it can lead to permanent damage of the kidney. Now, one way of treating this is to administer the person with ethanol, okay, which is an alcohol. So ethanol is an inhibitor to alcohol dehydrogenase. Okay, so ethanol is your inhibitor. Okay, and it's a good inhibitor because it can stop the production of oxalic acid. Okay, so ethanol is an inhibitor. Now, ethanol competitively inhibits alcohol dehydrogenase, so give the patient a massive dose of ethanol so as to prevent ethylene glycol from interacting with alcohol dehydrogenase. So the effect would be treating the person, okay, from being poisoned by ethylene glycol. So that is the effect, okay, of ethanol being an inhibitor. All right, so let's look at Ruslan and Shakruk's work. Snake venom. Okay, so inhibitor name is fasciculation. Very good. How does the inhibitor work? It acts as a competitive inhibitor, preventing acetylcholine from being broken down by acetylcholinesterase. So actually, this part is still how does the inhibitor work or the mechanism. So it's an inhibitor to acetylcholinesterase. If you guys can still remember, acetylcholinesterase is the one that breaks acetylcholine, okay, which is a neurotransmitter in synapses. And acetylcholine normally needs to be broken down, okay, so that the muscle can relax. Because if the acetylcholine is permanently bound to the receptor, that can lead to a prolonged contraction of muscle, which is not good. So you need to break down acetylcholine, okay, using acetylcholinesterase. However, if a person is bitten by a snake which contains the venom, that inhibitor fasciculation will inhibit acetylcholinesterase. So therefore, it can stop nerve impulses from being transmitted and hence stop muscles contraction. So this is correct. So that can lead to flaccid paralysis. Okay, so Shakruk, is this a positive or a negative effect of inhibitor? Fasciculation. Is this a positive or a negative inhibitor?
Positive for who or for what?
Is it positive or a negative for the person, of course, who's bitten by the snake? Is it positive or
For
negative?
human is positive. No,
Oh,
negative. But
negative
maybe
for
snake
the
is positive.
positive or negative?
Negative.
For human
Negative.
is negative. Yeah.
Yeah, for human it's negative. We're talking about the effect in humans. Okay, Mohamed Yar and Mahinur.
Yes.
Okay, so yours is protease inhibitors. So the name of the inhibitor is protease. Okay, and then How does the inhibitor work? The virus uses this to cut viral RNA into smaller pieces, so as into implant genes into the host cell's DNA and hence replicates. In viruses, they make use of enzymes, okay, protease, in order for them to replicate or reproduce themselves. Okay, so what happens is if a person is infected with HIV, that person is treated with this drug, okay, which contains the protease inhibitor. So that protease inhibitor will not allow the protease, okay, to reproduce, or that will not allow the protease to become active, which allows the viral DNA to be reproduced. So effect is a host cell can be infected by HIV, but it cannot be hijacked into making more HIV copies as a result of DNA implantation by the virus. So Mahinur, is this a positive or a negative effect of inhibitor?
Positive.
Okay, positive. This is a type of inhibitor that is found in drugs. Okay, so for a person who's infected with HIV, so this drug is being administered to that person so that the viral DNA or RNA will not be replicated, so they cannot reproduce. Okay. Last, Abdukuhur and Alpamas, cyanide poisoning.
Yes, Mr. Leo. Can you
Okay.
check?
All right. So cyanide poisoning, cyanide is the name of the inhibitor. Yes. How does the inhibitor work? Cytochrome oxidase normally combines oxygen and hydrogen together to form water and allows ATP creation. Okay, so the inhibitor, potassium cyanide, it inhibits this enzyme, the cytochrome oxidase. Okay, now cytochrome oxidase is one of the enzyme in the electron transport chain. Remember, in cellular respiration, during ATP production, most of the ATPs are produced in electron transport chain. And one of the enzyme there is cytochrome oxidase. Okay, so if cytochrome oxidase is inhibited, then the cell will not be able to produce ATP, okay, enough ATP. So any reactions requiring ATP are no longer supplied. Very good, okay. Remember, in cellular respiration, 2 ATPs are produced in glycolysis, 2 in Krebs, and 34 in electron transport chain. So this cytochrome oxidase is used in the electron transport chain. So imagine the 34 ATPs will not be produced if this one is inhibited, okay, by potassium cyanide. So the cell will not be able to produce enough ATP. Okay, now is this positive or negative? Abdukuhur. Positive
Yes,
or is this positive or negative effect?
it's negative effect.
Okay, definitely it's negative because of the word poisoning. Okay,
Yes.
so the first two, the snake venom and cyanide poisoning, are examples of negative. And then the last two, which is about protease inhibitor and ethanol, they are both examples of the positive effects of inhibitors. So not because inhibitors decrease the rate of reaction would mean that they are always negative. So there are also positive effects of inhibitors. All right, any question,
No
guys?
question.
Okay, so if no question, you have 1 minute break. Okay, let's just have 1 minute break, and then in the second lesson, we'll talk about the applications of enzymes.
Yes, guys, I won't say about it was very good job, but Mr. Leo, can you prepare a video? Yes, can you open?
Yes, yes.
Okay, thank you.
But first, I want to show them the assessment criteria because they're going to assess their classmates' video, right? So
Yes, yes,
every
sure.
video, they will assess their classmates' video.
Yeah,
We're going to ask questions as well.
yes, sure. I think, guys, we can continue our lesson.
All right, so please remember these 5 things that you need to assess your classmate. Okay, so you should be giving 5 points for your classmates' video or work. Okay, so the topic is about application of enzymes. So discuss possible use of enzymes in medicine, chemistry, and industry. And then we have the different applications of enzymes listed here. And you guys were asked to make a video presentation about the different applications of enzymes. So we're going to watch them one by one. And then after after video, you're going to assess your classmates' video, and we will also give some questions to you. Okay, so
Guys, do you understand
let's
what we
start.
will do? Do you understand our instruction?
Hello, my topic is beta-lactam.
Okay, let's start with Medina's video. Okay, everyone, please pay attention. Abdul Kohor will be giving his assessment to Medina's video.
Where
Hello,
is assessment
topic
criteria?
is
Sorry.
beta-lactams, and I
Stop,
want
stop,
to talk
Mr.
about.
Leo. Mr.
Yeah.
Leo, can you
Uh-huh.
show again criteria, success criteria, because Abdul Kahor asked again what assessment criteria.
Here. Okay, so creativity, does it discuss the application of the enzyme? The language is of the video is fluent, understandable. The content is clear and structured, and it makes use of technical terms related to enzyme. Okay?
Can you take
Okay,
photo,
okay.
please, guys? Okay, and you can use when you will assess.
Already done. I sent it in WhatsApp to our group.
Okay, thank you. Okay, we start, guys.
So, Abdul Kohur.
Hello, my topic is beta-lactams, and I want to talk about the structure and use of this enzyme. So let's start. Lactam is ethyl-gametes, and beta-lactams are named so because the nitrogen atom is attached to the beta-carbon atom relative to the
Mr. Leo.
Я думал, это у меня зависло.
Mr. Leo.
can see. The common thing is that all these groups have beta-lactam ring in the center of structure. So beta-lactam antibiotics are indicated for the prevention and treatment of bacterial infections caused by susceptible organisms.
Okay. Abdul Kohur, what can you say about Medina's video?
Okay, I think Medina create a good video content. However, there are some mistakes. I think that one criteria is haven't done because there are 47 seconds. Medina should create a video with 60 or 90 seconds.
Okay, yeah. So the video.
Also,
go on.
Also, language was clear and I understood all what she said, and she used some technical terms like beta lactate, nitrogen bonds, or and so on. That's all.
Okay, yeah. So I agree the video is short. Okay, so she could have explained more about beta lactamase, because she only mentioned about beta lactam, which is a chemical found in antibiotics, which is used in killing bacteria. So she mentioned about penicillin, cephalosporins, okay, but she didn't mention about the beta lactamase, okay, which is the enzyme that bacteria produce when they become resistant to this antibiotic. Okay, so beta lactamase is actually a bad enzyme, because this will allow the bacteria to become resistant to antibiotics. Okay, so now let's watch the second video from Abdul Kahur, and we will ask Ruslan.
Hello.
Okay, we will ask Ruslan to assess the video.
Hello everyone. Now I will tell you about application of pectinase enzyme. Pectinase have a variety of application in the industrial sector. This particular enzyme is used in the extraction of fruit juices such as apples, strawberries, grapes, raspberries, etc. Likewise, pectinase plays an important role in industrial processes such as plant fiber damming, water treatment, oil extraction, coffee and tea fermentation, and the paper and pulp industries. Fruit extraction occurs when the pectinase enzyme breaks down the pectin substrate and the juice is extracted. The enzyme pectinase reduces the activation energy needed to produce juice and catalyzes the reaction. Also, pectinase are applied for the treatment of wastewater coming from different industries. Textile processing and bioscoring of cotton also find a role of pectinase in their processes. Pectinase is also used in paper making. It has the ability to depolymerize pectin. That's all.
That's
Okay,
all.
Ruslan. Ruslan?
Yes, I think that overall the video has high quality, except of the approach to present information to us, because we just saw the Abdul Kahor's face, and I think that we need some pictures or schemes to remember this. But in other aspects, he spoke clearly in English language. Also discussed about enzymes application clearly using needed terms, but I think that to remember these terms, we need to see it, but not to hear.
Okay, good, Ruslan. I agree with your feedback. Abdukahor, next time when you will create video, please use some pictures or some simulation for explain your topic. Okay.
Yeah.
Do you agree, Abdukahor? Do you agree?
But for me, yes,
Yes, I absolutely agree.
Okay.
The voice is very clear that I understand what the application is about. It's pectinase, which is used in fruit juice production. And he did not only mention about the use of pectinase in fruit juice production, but in also other industries. So that's good. Okay, again, just add a little bit of visuals to make it more creative. Okay. All right. Next, Ruslan's video, and Mahinur will give her evaluation. Okay. So Mahinur will assess the video of Ruslan.
Topic is enzymes used in alcohol. I took two parts when we use enzymes. Firstly, it is alcohol fermentation, also a chemical breakdown of alcohol. Firstly, my first enzyme is invertase. It uses in making glucose. Its main function is
Mr. Leo, your microphone.
Alcohol dehydrogenase to make ethanol. Their function is to catalyze these reactions. So I wrote it here, these enzymes is a catalyzer. Here enzyme is catalyzer. The second part is chemical breakdown. To get acetate from ethanol, we use two enzymes. First is alcohol dehydrogenase. We can write it as ADH. And aldehyde dehydrogenase. It just changes the molecular structure of ethanol to get acetate as a result.
Okay, so Mahinur, what can you say about Ruslan's video?
Okay, it was really good job, but the only minuses are is that camera was always moving and it's sometimes creates difficulties in understanding, and also I would suggest him to use presentations or more precise handwriting.
Okay. Do you agree with that, Ruslan?
But it was a good job. I like the way that he used formulas.
Yes. Okay. Now that's good because he presented all the formula, okay, needed for alcoholic fermentation. Okay. So for someone who understands chemistry or who studies chemistry and biology or biochemistry, so this is very clear. Okay. But for someone who doesn't really understand chemistry, this could be quite difficult to understand. So what I can suggest is to put some pictures again, because visuals and pictures would really help people to understand what you are presenting. Okay, but again, it's precise, accurate. So you've mentioned about the use of starch at first, which will be broken down by the enzyme invertase to produce glucose, and then this glucose will be used by microorganisms to do alcoholic fermentation, okay, which in the end will produce ethanol, which is an alcohol. So alcoholic fermentation is used in beer production, in brewing beer, okay? Yeah, so again, it's just that the camera is shaky and you could also improve your handwriting. Okay, thank you, Ruslan and Mahinur. Now let's go to Mahinur's video. And the one who will evaluate her video is
My
Mohamed
topic
Yar.
is
So Mohamed Yar will assess the video.
immobilized enzyme. An immobilized enzyme is an enzyme attached to an inert insoluble material such as calcium alginate. Immobilized enzyme systems. Enzyme immobilization is done in order to restrict enzyme mobility in a fixed space. Materials to which enzyme attach will prevent loss of enzyme activity by not changing chemical nature of the reactive groups in the binding site of the enzyme. Here are some main disadvantages and advantages of immobilized enzymes. The main advantages are enzymes can be used for several times. It is stable for high temperatures and pH. Disadvantages are immobilization may alter enzyme shape and function, high price of immobilizing materials, and enzymes may detach. How are immobilized enzymes used in industry? Immobilized enzymes are utilized in a wide variety of industrial practices. It can be used in production of biofuels, paper, and polishing clothes. Why are immobilized enzymes more stable? The increased stability may be a result of the fact that immobilization limits the thermal movement of the enzyme at the higher temperatures. As a result, thermal denaturation may not occur at higher temperatures with an immobilized enzyme.
Okay, Mohamed Yar.
Mohamed Yar. Mohamed Yar.
I think he has some problem with his microphone.
Microphone.
Like I can hear, but it's very low.
Okay, Mohamed Yar.
Okay, so
Like,
he was actually saying his assessment.
yes.
It's just that maybe there's a problem with the connection, so we can't hear it very well. Okay. All right. So.
But my suggestion just for Mahinur is when you create a presentation, you must use just short text, not a lot. For example, you show one slide, it's a lot of information which I must read, but I will not read, yes. Please, maybe you can use some pictures which show what you want to say.
Oh, yeah.
It's my suggestion, Mr. Leo.
Yes. Well, the idea of immobilizing an enzyme is to make the enzyme reusable. Okay, because enzymes are actually expensive. Okay, so by immobilizing the enzyme, we can actually reuse these enzymes. Okay, for example, production of lactose-free milk. Okay, so lactose-free milk are being produced using immobilized enzymes. And another good thing about using immobilized enzymes is that they are more stable. So they can last longer compared to the normal enzymes, okay, or the free enzymes. All right, so now let's watch Mohamed Yar's video. The one who will assess this video is...
Like any other living materials, the cell...
Okay, Shakruk will assess Mohamed Yar's video.
Like any other living materials, the cells of cereals uses for the production of flour contain enzymes, of which amylase and proteases are the most important. However, the amount of these enzymes is not available to baking, and sometimes it's necessary to add additional enzymes. Enzymes preparation are catalysts for biochemical processes occurring in organic compounds. During the preparation of bread and flour production, enzymes are inactivated and there are no finished products. Enzymes are introduced in dry or dissolved form when kneading the dough. The enzyme solution is stored up to 24 hours. Using natural supplements, enzymes preparation that have complex technological effect and create opportunities to quality improvement. Differential products of flour for bread from a mixture of rye and wheat. Mass varieties of bakery products, flour confectionery products, crackers, cookies, biscuits, pastry, and so on. Pasta production of ready-made mixes for special purposes. Stability of working with the products of flour. And last one, increase the economic efficiency of products.
Okay, Shahrukh, what can you say about Mohamed Yar's video? So he's talking about the use of enzymes in baking, bread making.
So he described all information, all which we need.
Okay.
But he should add pictures or some schemes, not only show his face.
Okay. Yeah, it's actually good that we're able to see you. Okay, because in quarantine during our lessons we don't get to see your
Additional
face.
recommendation.
Okay, Shakrut, go on. Yeah,
But I like Muhammadyar's emotions,
Yeah.
how he spoke.
Okay, so it would be
Mr. Leo.
better if there would be again pictures, visuals that you can show, even though yes.
Can you show this second video, not the first?
Okay, all right. So let's look at Shahruk's video.
Not. No, I
Shahrukh,
sent
Shahrukh,
video.
Shahrukh.
Yes,
This
yes,
one.
yes, that.
This one.
Yes,
Right?
yes.
Okay, all right. Okay. So Alpamas, please assess his video.
People have been experimenting with this to use the power of enzymes to clean clothing for a long time. In fact, the first application was in 1970. And because of stains are made of different types of molecules, a range of enzymes are needed to break them down. The first is protease. Proteases break down protein chains, so they are good for eggs, plus, and gravy, and other protein stains. Also, amylases break down starch, and lipids break down fats and oils. And we can find in washing powders these types of enzymes. And advantages of using enzymes in washing powders that dirt and stains can be easily removed without the need for hot water. Also, we can pop a wider variety of materials into the washing machine, such as wool and silk, which could become damaged in high temperatures. Also, dyed clothes. Enzymes reduce color transfer in washing machine. So there are benefits of using enzymes in washing powders.
Okay. Alpamas, very quick assessment.
Very informative video, and video timing is also acceptable.
Okay, and he presented it in a way that ordinary people could understand. Okay, even though he did not use any pictures, so he used like bullet points, okay, to outline the use of enzymes in biological washing powders. Okay, for example, you have a blood stain or chocolate. So chocolate contains lipids, so the biological washing powder should contain lipase so that the chocolate stain can be removed. Blood contains protein, so the protease enzyme can break down the protein into amino acids, which are which can be dissolved in water. Okay, so it's good. And he also mentioned about for dyed clothes, so there are enzymes which can reduce color transfer. So for example, you have two clothes, one is blue, one is red. So there is an enzyme that will prevent the mixing of the colors, okay, between the two clothes. Okay, so for me, this one is a good one. You just need to like be more livelier in the way that you talk.
Can we finish our lesson? Yes, Mr. Leo.
Yes.
Okay. Goodbye. Goodbye,
Goodbye.
students. Thank you very much.
Have a nice day. Goodbye, everyone.